Kia ora and welcome to our June LINKOnline webinar. I'm Christine Harper, Manaaki Whenua's Stakeholder Relationship Manager, and the best part of my job is helping to join the dots between our research and the questions that our stakeholders are grappling with. More Birds in the Bush is a large, collaborative, MBIE-funded research program, and we're at the midway point where we're already improving the ability to forecast predator threats and bird responses and making an impact on the effectiveness and efficiency in forest management nationwide. Susan Walker and Adrian Monks are senior ecological researchers at Manaaki Whenua in Dunedin, and they're about to provide a condensed version of 2 full days of presentations in just 20 minutes. I'll be back at the end of the presentation to manage our question and answer session. Don't forget to put your questions in the chat box as soon as you can. But gosh, 20 minutes, 2 days work. Over to you, Susan and Adrian.
Thank you, Christine. Tēnā koutou katoa, and thanks to everyone who's come along. We're really looking forward to your feedback and your questions. Oh, my screen's not advancing. There we go. Okay, first to say thanks. We're fronting for a very big team today, and we've had a ton of behind-the-scenes support right across the motu. We also acknowledge MBIE-funded and the sharing of people and data by our big organisation partners. Today, we're going to start with just an overview, and then we've decided to focus on 2 brief takes of our results, both focused on ship rats, to dive in a bit deeper there, and we're going to finish up with where the program's heading to now. We'll direct anyone wanting to dig deeper to the sessions and the presentations from our recent 2-day mid-program webinar that Christine mentioned. They're up on our website. So our research aim is to create the ability to predict threats and forest bird responses across all of New Zealand's forest types so that people can better restore those forests and the populations of forest birds in them to health by developing management approaches that meet their aspirations for the forest and are also better for birds. Our research focuses on large remaining forest tracts, and it has a big emphasis on gradients in temperature across those forests. So warmth decreases, as you can see in this map, from north to south, and also as you travel across our islands from east to west. Our coldest forests are simple. They've got relatively few species, and they've got relatively little food, and they have few predators in them. Warmer forests are richer. They support a much greater diversity of species, they provide more food, and they should support bigger, healthier populations of forest birds. But today they don't, and in large part that's because they have more predators. And so our warmer forests are both a great problem and a great opportunity if we care about more birds in the bush. Now, as our research has been progressing, we're confirming how few forests truly have cold forest character. In our research proposal a few years back, we used 16%, but that number's a real moving feast as climate warming is progressing. So warmth and the food resources that are associated with that warmth favour the predator that's the major focus of our program, the ship rat. And this is a predator that's very hard to control and manage, as a lot of predator-free projects are finding. To be able to predict and to manage ship rats and birds, we need a much better handle on the food in the forests, flowers, fruit, fungi, insects, which drive their populations. And so this is the first theme in our big program. We also need to know how effective our management is at enabling populations of forest birds to survive and thrive. So the second big theme is predators and management. And finally, we need to know how birds in the bush respond to all those things, to food, to predators, and to our management in these big forest blocks. And our mid-program update has blocks of presentation in all those 3 themes, and they're up on the web. Now, over the next couple of years, we're going to be working alongside our partners even more to help them achieve their goals through a set of case study applications. But for today, I'm going to hand over to Adrian to discuss some science results, first from a very big field experiment in a very big beach mast. Over to you, Adrian.
Thank you, Susan. So I'm going to talk to you a little bit right now about what limits ship rats in cold forests. So this work was led by Joe Carpenter with some input from quite a sizable team. So the motivation for this work really was the observation that a lot of rat-sensitive species are increasingly becoming rat-sensitive bird species are increasingly becoming confined to high elevation forests, and that as a correlation of that, ship rats are generally less abundant in those forests. Now, there's two main ideas about why ship rats might be less abundant in those high elevation forests. One is that there could be some sort of direct temperature limitation. You know, they are physiologically ill-suited to survive through the winters in those colder environments. But the other aspect of this is that those forests tend to be much simpler floristically. And so it may just be that there is less food available for them. And so the primary mechanism here is about food limitation. Next slide, please, Susan. So this study was set, sorry? This study was set up at Lake Alabasta in Fiordland, and we made use of a large beach mast in autumn 2019. Now, the idea behind this is that in these cold forests, rats are mostly fairly absent, but we do know that they tend to have large population abundances in the spring and summer following large mast events. So the program started in October 2018, and then we suddenly were confronted by this large beach mast where we had to get a study up and running. And Joe was only brought on to undertake this work in January in 2019. So it was actually a massive effort to get this going and actually have boots on the ground and people measuring rat abundance by winter of 2019. So one of the problems with that is that we've kind of missed the early part of the population increase with the mast. So as I was saying, the study ran from July 2019 right through to January 2021 over a beach mast cycle. There were quarterly ship rat density estimation at 3 elevations from 20 meters up to 900 meters. So we use capture mark recapture, which involves capturing rats, releasing them, so this is live capture, and then trying to capture them again. And that sort of gives you information about how far they're moving across that landscape, and also the likelihood that you are to observe them. And from that information, you can fit some fancy models and actually estimate density. One of the things we specifically wanted to test was that once the beach mast is gone, you know, the seed is no longer available, can we keep those rat populations high by supplementary feeding them? So in two of those high elevation grids, we supplementary fed them using sheep nuts in a bait station grid from January 2020, which was at the end of the availability of the mast, through to spring of 2020. And at the same time, we also indexed stoats and seeds and invertebrates, which is just food for rats. So I just want to talk you through the main result from this, which is really what the populations did at that site. So on the top graph, we have rats per hectare, and the bottom one, we have detections of stoats. The grey area that you can see shaded there is the availability of seed. So the seed is falling during March in 2019, and then it's lying on the ground, and then as it gets into spring, it starts to germinate or rot or is eaten by rats. And so by about January 2020, we no longer have any seed in the system. Now, as you can see there are 3 elevations of rats. So the red one refers to high elevation rats. The light blue one refers to low elevation rats, and the gold one refers to mid elevation rats. And then you can also see that there is a dark blue spots, and that relates to our supplementary fed grid at high elevations. Next slide, please, Susan. All right, so I want to focus first on this first period. So as you can see, rats during this initial period, by the time we actually started sampling, were already really high, and these are some of the highest densities that have been observed anywhere. We don't really know whether this is kind of typical of these systems because there haven't been that many density studies, but these are high numbers of rats at mid and low elevations. But what you'll notice from that is at the start of the study, rat density was really low at high elevation sites. Like we only caught about 2 rats during that first sampling period. And so during that period, they slowly increased. And so there's a lag in there between what was going on at mid and low elevations and what was occurring at high elevations. And this is kind of quite important because it means that because they had that kind of late start, they never quite reached the same high levels of density as what the mid and low elevation grids did by the time the food ran out. And one of the things that we've shown with some fancy modeling is that this increase at the high elevation sites is completely dependent on there being some form of immigration from those lower sites. So without that, just from breeding from in situ rats, we would expect that density to be about 1.8 rats per hectare, whereas in reality what we observed was it was from 6 to 10 rats per hectare. Next slide, please, Susan. Okay, and this is the decline period. So by this time the beach seed has run out, there's high densities of rats. The rats are in really poor condition, indicating that actually they're struggling for food. So they've eaten all the seed resources. Invertebrate densities are quite low at the same period of time. And so we're starting to see a decline at all elevations. And as you can see here, we set up our supplementary feeding grids to just pick up the end of that beach seed availability and just to see if we could kind of maintain populations through that time. You might also notice in the bottom panel that stoats are beginning to increase in this time as well. They weren't very high initially when that decline first occurs, but by the end of that period, they're actually quite high. And I should note also that the mice are fairly high throughout this period as well. Next slide, please, Susan. Okay, and the last thing I'd like to sort of point out was the effect of the supplementary feeding. So if you go down to that bottom right corner, you can see that that dark blue line, the actual rate of decline of the population was much slower in our supplementary fed grids. And it's not that it didn't go down at all, but we managed to slow it, and it's significantly different. So over that wintering period, there were more rats on our supplementary fed grids than what there were on non-fed grids. But by the time we get to spring in 2020, they have all crashed. And the likely reason for that is because the stoats are still eating them. And so they're still steadily going down and down. Next slide, please, Susan. So in summary, we have shiprat eruptions at high elevation contingent on this immigration. So that's one of the key findings, I guess, from this study is that there aren't really enough rats in the system at high elevation to actually cause an eruption of rats. So this sort of raises the possibility, well, if we could somehow prevent that, you know, maybe we could without having to drop poison at high elevation, for example, manage to limit the populations of rats and therefore the damage that they're doing up there. But that also comes with some risks because it means that you also have to keep densities in those low and mid elevation grids low for a long sustained period of time to prevent that kind of building up of the population and allowing that immigration to happen. The decline is caused by food limitation and definitely hastened by stoat predation. And so it really looks like the direct impacts of temperature, you know, that physiological limitation are really trumped by baseline food availability. And that's what's driving our low densities of rats at those high elevation sites. Okay, Susan, thanks.
Thanks, Adrian. I'm going to talk now about what we've discovered about shiprat patterns nationally. And this is work by Docs Josh Kemp and Graham Elliott, along with Rochelle Binney and me, and we're trying to understand how shiprat tracking rates, which are an index of rodent activity, they're not the fancy mark recapture stuff that Adrian talked about, are affected by aerial management and ecosystem productivity and by the different management regimes or control cycles we use in our remaining large forests. This is to help us predict outcomes of management for birds across all our different forest types. So we use the the huge national data set of overnight tracking, which is mainly DOCS efforts, but acknowledging also we've got some Greater Wellington records in here. The data go up to August 2020, at least the ones we used, and that's the winter just following the big rodent explosion that Adrian just covered. Three things: the tracking network grew over time. We used the 16 years when coverage became seriously large. Second, there's far more data available for the South Island forests than for the North. And third, those data cover three main types of forest: the pure beech, the mixed beech, and the podocarp broadleaf forest as we've divided them, which allows us to say something about these areas of forest, again in the three types. And a key point in all of these forests and across those forests is that forest type and temperature are not independent. So New Zealand's coldest forests are exclusively the pure beech forests at the left, and the warmest are podocarp broadleaf. In this work, we divide pure beech forests into 6 classes, each with a 1 degree temperature span, and the pure beech go from 5 to 11. The mixed beech goes from 7 to 12, and the podocarp broadleaf, our warmest forests, go from 9 to 13. And these are some results for those forests. So these are shiprat tracking rates across all of our forests if we do nothing at all, shown quarterly from November 2005 through to 2020, 16 years. For the 15 forest temperature classes, which are shown in different colors in the legends down the right-hand side. And you can see immediately how shiprat tracking rates increase substantially with temperature within each of those 3 major forest types. The deeper oranges and reds at the top of those graphs, each panel, are our warmer forests. And this instantly explains why smaller species, native species that are vulnerable to shiprats like mohua, robin, titiponamu, kakariki, and bats are already long gone or very rare in New Zealand's warmer forests, and why those cold pure beech forests are their thermal refuges. The pure beech forests at the top, in the top panel, are now the main refuges, but shiprats are not always rare there. Those arrows in the top panel show the rat eruptions that followed beech mast seeding through that period, and the low tracking rates between them are what we call intermast periods. A feature of concern is that base tracking rates of shiprats have been rising across New Zealand forests over the last 2 decades, including in those pure beech forests that are the thermal refuges of many birds. And you can see in this slide the distinctly higher shiprat tracking rates during the last intermast, the one labelled post-2014, which temperature records show was the warmest intermast in history, and rats are much more tracking much higher in our forests than before. So climate change is not our friend if we're trying to tackle shiprats. So moving now to what happens when we do aerial management against all that background pattern. So again, we've got median tracking rates predicted by models on the vertical axis, but on these graphs, the horizontal shows the years since any aerial management operation undertaken since 2005, which is recorded in the data set. And what we see is that aerial management immediately knocks tracking rates to 0 or very low levels in all forests. It does a really good job, but then tracking increases and it reaches new maxima between about 2 and 3 years after an operation, and then it recedes again down to the long-term median. So initial increases after management are more gradual if you look near the top in our cooler forests, but they're almost vertical in those warmer forests. Shiprats just go crazy when you've knocked them down with aerial management in those warm forests. So in this slide on the right, I've put the unmanaged rates, the if we did nothing scenario that we can compare those managed rates to, and we get the difference made by aerial management if we subtract the unmanaged on the right from the managed on the left, and that's what I've done in this slide here. And you can see the striking pattern in the difference that aerial management makes. So here, dots below the line is good. Shiprat tracking rates are lower than if we did nothing. But these humps here above the line in all our forests show the rat bounce, the period of time 2 to 5 years after an operation that shiprat tracking rates become higher than they would have been if the forest had not been managed at all. And these horizontal lines show the significant time periods of unders, and these ones show over with the thick lines showing statistically significant differences. So all forests a significant initial knockdown. In pure beech forests, we have later, smaller, briefer rat bounces, almost negligible in cooler classes, but in warmer mixed beech and in podocarp forests, we get these larger, longer, and more significant rat bounces. And our analyses tell us this rat bounce is largely due to food. We make forests better by killing the omnivorous rats and possums that strangle forest food supply and eat birds as a side dish, but ironically, ship rats then benefit from the food bonanza, especially in warm forests. A smaller but not negligible contributor to the rat bounce is that aerial management achieves excellent and longer-lived suppression of stoats, which are a key predator. So rats get a window of relief from persecution as well as more food. The panel on the right shows those much longer windows of suppression we get in mustelid tracking rates, at least 2 years of relief in all types of forests. Stoats also bounce, but it's much later. So together, what this comes together to mean is that the outcomes of aerial management are likely to be much better and much longer lasting for larger bodied species, kaka, kea, kiwi, and whio, that are vulnerable to stoats, but not so much to rats, than for the smaller mohua, robin, titipunamu, kakariki, bats. And as I said, those bird species and bats are already mostly gone from our warmer and rattier forests, but because of the rat bounce, we can't rely on infrequent aerial management to allow them back in. And the other points I made earlier in my talk are that climate change and ground-based methods of stoat control, such as trapping, are also likely to favour ship rats in our forests, which confirms their status as that really formidable and challenging predator to control. I'm going to hand over again to Adrian.
Thanks, Susan. So next slide. Cool. Okay. So one of the key places that we want to get here really is, how does all this matter for outcomes for birds? And this turns out to be a surprisingly hard thing to do. You know, like, it's really difficult to measure outcomes for birds just because they're so difficult to work with. What we'd like to develop is spatially explicit management prescriptions for bird outcomes, and we want to integrate this with our knowledge of bird life histories. I mean, Susan's already alluded to the fact that with the rat bounce, some rat-sensitive species may actually suffer a little bit more under 1080 ops. So understanding sort of when, where, and how to use the management tools that we have is kind of key to our next steps. We also want to understand how management under current and future climate might change. It's sort of a key thing, obviously, with a warming climate. And the other thing that's kind of on our radar is how management might change under harvesting regime. So, you know, obviously now some populations are harvested for translocations. In the future, there may also be cultural harvest as well. So that's something that's also on our radar. Next slide, please, Susan. And so how are we planning on doing this? Well, as Susan's already alluded to, there are a whole bunch of different strands to this program, and we've only kind of shown you a very small part of it here. What we're planning on doing is integrating this in a big broad scale model that brings together a whole bunch of different strands of research. So there's the food productivity one, how that changes with space and time and environmental change. There's rat dynamics, as we've talked about today, but also the effect of those rats on the availability of food. How both of those things affect the vital rates of the birds, you know, survival and fecundity. And so built into this will be a population model. And then how all of this kind of comes together within all these various management scenarios of, you know, perhaps pest control or translocations or what have you, to generate more birds in the bush. Next, please, Susan. So you can find a lot more detail on some of the things that are undergoing that we've done in our program in our recent mid program meeting. There's a whole bunch of different things in there, and it's well worth sort of poking around. You'll get a lot more detail than what Susan and I could offer you today. Yeah, I think that's about it. So thanks very much for listening, and yeah, we'll take some questions.
Thanks, guys. There's some great questions coming through as well, but I've got one first. Why are you working with ship rats?
Shall I answer that, Adrian?
Yeah, go for it.
So ship rats are our forest rat. They're also a very worthy enemy. Like they are a species that really we have, I guess, fewer tools for, especially at large scales. Yep, and they're a keystone species because they not only eat birds, they also we're learning more to control forest food supply. So it may be that our cold beech forests are actually controlled by ship rats and by mice, you know, that they have little food because everything gets scooped up by them. And of course, they feed stoats. And so they're a major bottom-up driver of those bigger predators as well. They're just at the center of that forest predation food web.
Thank you. Great context. Question, I think this one's for you, Adrian. But how did you distinguish immigration from recruitment at high elevations?
So with some fancy modelling, so we ran some population models to try and predict what the densities were, the observed densities that we had at these sites. And in those models, we included immigration terms for movement from populations lower down the mountain to those high elevation sites, and then compared that with what would happen with a model where you didn't have any immigration built into that. And so for reasonable amounts of recruitment, you couldn't attain the densities that we have observed in the field.
Thanks. Now, I think this one is for Susan. Is the research showing that no management is better in a podocarp broadleaf forest than any management due to the rat bounce?
No, I think that's you do get your breeding season for birds out of that no matter what. But the other thing is, I think what it's telling us is that when you're doing management, especially in those warm forests, you have to think about what you're managing for, because if you do nothing, your large birds are going to get eaten. If you've got kōkako, you have to think about when the rats come back. You're achieving great suppression of possums usually, and so it's much more complex than an either or, don't do anything. Small birds are going to be much harder, and so that's, I guess, what the real sort of tailoring of management is going to have to grapple with. Big birds are kind of easy because 1080 does a great job on stoats, and that's all you really have to worry about. But there's a lot more to worry about in warm forests if you're trying to maintain small bird species that are rat sensitive.
Right, and just picking up on that small theme, did you track mice, and to what extent are they also a food resource?
Mice are definitely a food resource for rats, and yes, we did, and no results on that here, but yeah, that interplay between them is fascinating. And yes, if we do control rats successfully, which is still a challenge that I don't think we've met, mice will definitely be the beneficiaries of those. And so, yeah, it's thinking about those forests as this whole food web, and everything you do has another reaction somewhere. Yeah.
Right. We have got some more questions, but we are running out of time to get to all of them. Some of them are really complex, and I know they're not easily answered. We will come back and answer those questions. So thank you to everyone who has put them in. Still a chance to fire in a last question, and you will get a personal response from Susan and Adrian, and we'll also post those to our website along with the presentation afterwards. You will also have an email from us with a recording for the presentation, and this is really a good time for us to be talking about birds, and for you yourself at home to contribute to our bird research from this weekend. So the Garden Bird Survey starts on Saturday and runs for a week. You don't need to go to the forest. Just find a local garden or park and put aside an hour of peacefulness to go and sit in the garden and count the birds that you see in that hour. There will be a link as well as to this presentation, but also to the Garden Bird Survey and to the full gamut of presentations from our earlier workshop on this work. We'll be back with another webinar in July, so watch your inbox for information on that. And I'd just like to finish off by saying a huge thank you to Susan and Adrian. This is fantastic, and we look forward to welcoming you back towards the end of the program for an update. Thanks, and bye for now.